And so I think we should get going. So good afternoon, everybody. Warm welcome also to all external colleagues and stakeholders who are joining the public session. So why do we have a public session? Quite simply because we feel that VetBioNet has to gain visibility. That's something that we have discussed earlier during the restricted session of the annual meeting. And this is basically an effort to showcase our activities. So for this public session, we have selected 3 important and timely topics. The 3 topics are transnational access, in other words, the services that we are providing, our core activity, the heart of our project. The second part will be presentations about COVID-19 research activities by VetBioNet partners. And the public session will then be finalized by presentations of our joint research activities in a partnership with private partners. So I will now let the screen, microphone, floor, whatever you want, to Norbert Stockhofe, who will present the first session about transnational access. Okay, go ahead, Norbert.
Thank you, Sasha. I will try to share my screen. I hope you can see it now. Just confirm.
Yes.
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 Okay. Okay. First of all, thank you for giving the opportunity to try to show the results of, in our case, 2 TNA projects. The TNA is standing for Transnational Access, and within VetBioNet, this is a central part of offering the expertise and the infrastructure to the research community, and this all for free. This is something which is really important. The idea behind the transnational access, and I'm talking now especially to those who are not within the within the transnational access activities and within VetBioNet, especially to the visitors from outside the consortium. Just to tell you that the transnational access is a free of charge possibility to perform animal studies with infectious agents, usually with BSL-3 agents, but with exceptions on with BSL-2 agents, as they might need a certain containment. And I also will take the chance to tell now that we still have a year to go to accept studies, and but this is under the assumptions that we will have an extension of the project for half a year, and we still have budget and capacities available to do so. Just to show you what has been done in the last I think it was nearly 3 years. We have projects from various countries in Europe, but also from outside Europe. It was that is a possibility within the transnational access, which is allowed by the EC on using your budget. And you see that we have a couple of studies from Spanish research groups, from Italian research groups, from France, Sweden, Denmark, Switzerland, but also from Cuba, Thailand, Japan, and Lebanon. And when we are going through the approved projects, which pathogens are involved, then you see a huge range of infectious agents, and you also see within this the competence we have available in the VetBioNet infrastructure to work with the different pathogens, but also to have the different pathogens available, and also to have the necessary laboratory assessments available. This is quite nice, and just this colorful picture shows how broad the scope is. And also this bar graph shows you a little bit on which groups of pathogens this were, and we were according to the One Health aspect of most of our institutes we are working in is on zoonosis, but the biggest part is on notifiable disease, and there are also some on BSL-2 pathogens as they require containment for doing studies, for example, with GMOs. And if we look for the also for the different host species we are working, I think we can be proud on what we can offer in the different species across various partners. And at the moment, we have 20 partners who contribute in various ways to the transnational access. And where studies can be performed, different studies, either animal studies, but also laboratory studies can be performed, or can be combined so that the study can be done partially at one institute, but as an assessment and analysis can be better performed in another institute. It could also be made possible. So this is a very short introduction on what has been done so far in the project, but also an example of what is possible with this type of infrastructures. And please take the chance or use this information to communicate this to your colleagues, that there is a possibility to accelerate their research if there is some necessity for animal studies or for certain specific studies which can be accommodated within the VetBioNet infrastructure. I'm really happy that we had 2 users of a project available to present their projects and to show what the studies went about, and also to give an insight in their experiences with doing the studies in an VetBioNet infrastructure. And I also am happy that the service providers are also available to give an insight in their experiences with providing such a service, because this also has impact on your daily work. The first is Maria Forlenza. I welcome Maria from Wageningen University Research, a colleague here in the Netherlands, and she did a study at INRA-IRP. And Bertrand Colette will hopefully be able to answer questions of the audience, but also we will have, I think, a short discussion on his ideas on the on providing the service. The second user, Ana Maria Moreno, she has to apologize not to be able to present herself the experimental infection of ewes and pigs with no Pestivirus, but Llilianne Ganges from IRTA, who provided the service, will take the opportunity to show the results. So, Maria, I'm happy to, I would like you to share your screen. I will stop sharing screen, and hopefully you will be able to share your screen and to present the your work on the mucosal vaccination of carps.
Just one comment while Maria is uploading her presentation. If you have questions, you can also ask them in the chat. So I will follow the chat. I assume Norbert also, and we can compile the questions also.
Yeah.
Let me see. Are you seeing something?
Yes.
Yes.
Excellent.
Let me see, because now my screen is very busy. Hopefully now is on the presentation mode. Yes, so thank you very much, Norbert, for the invitation, but also for the opportunity to tell a little bit about my experience with the INRA-IRP facility. And for this presentation, I had to make a selection of all the work that I was able to do at the facility. So I started a bit of a way back, but I hope I can tell you a little bit about our experience on successes, but also challenges on the development of mucosal vaccines against a disease of carp called spring Viremia carp virus that I will tell you a little bit more in a couple of slides. Let me see. Yes. So for those who are not so familiar with spring viremia of carp, it is a disease that, also given to the name, occurs usually in spring when the water starts kind of warming up after the winter period, and fish, of course, are not too let's say the immune system of the fish is not too awake, and that gives a bit of a window of opportunity to the virus to infect cyprinid, so not only common carp, but also other carp species. But common carp is one of the most affected ones. And indeed, basically in all countries, SVCV, so the virus causing spring viremia of carp, is among one of the notifiable diseases to the International Office of Epizoonotics, or OEA, which was also previously mentioned. And a little bit about the virus. It belongs to the family of Rhabdoviridae. It's a very simple genome, 5 open reading frame, has, for example, the rabies virus. And we already know from previous studies which one is our target for vaccination, which is the glycoprotein on the surface of the virion. We also know, based on, let's say, practice and farmers' reports, especially in East Europe and China, that young carp are the most susceptible, and susceptibility goes down, is reduced as the carp grow older. But there are some species, like, for example, koi carp, that still remain highly susceptible also at later stages. And that is why, of course, a vaccine is necessary, because although common carp might not be the most expensive fish, for example, when compared to trout or salmon, if you then also consider koi carp and the trade of cyprinid fish all over the world, the spreading of such a disease all over the world can be very dangerous, because it may lead to eradication of the farm or of the koi carp if the koi carp is infected. And considering that these koi carp are extremely expensive, then if you could vaccinate your carp before transporting it, it would be quite advantageous. So this is why we've been putting quite some effort over the years in trying to develop a vaccine against spring viremia carp virus. We started with the development of a DNA vaccine, and slowly we are trying to turn this DNA vaccine from an injectable vaccine to a mucosal vaccine that can be given either orally or by immersion, and that's what I will show you in a few slides. So indeed, how did we over the years start and progressed? So I actually started collaborating with the facility already within the AquaXcel project, and now more recently through the VetBioNet opportunity. And when we started, we had nothing. So we did not have a vaccine candidate. We did not have a optimized challenge model. And as you can imagine, that is quite crucial, because to be able to evaluate your vaccine, you would like to, of course, expose your animals to the pathogen in the most natural way. So we had to, of course, optimize a challenge route that was not only depending, for example, on injection, which was the most common route obtained or used up until now, but, for example, by immersion. We also, of course, optimized the pathogen dose. We had to look at our window of susceptibility, because, as I told you, young carp are more susceptible, and then as they grow up, they may become naturally resistant. But of course, we cannot vaccinate them too young, because, of course, their immune system is not developed. Plus, we had to come up with a system to be able to not only evaluate the vaccine ad intra, but also to be able to correlate protection to immune parameters, and that was done in Wageningen. So hopefully in the next slides, I will show you how we managed to combine and solve all these important points, and to come up with some successful vaccines. So first we started with the bat challenge. Already then, we managed to, so we have in Wageningen a excellent fish facility where we can grow SPF, specific pathogen-free carp, common carp, of a particular strain that is, let's say, an outbred strain of carp, R3R8. And these were basically bred in Wageningen, and the eggs within a day were shipped immediately to Indra. Fish were grown for 3 months, and then at Indra, they set up an experiment to test both intramuscular and intraperitoneal challenge using a virus that was also provided by Indra, so the Canclen strain. And here you can already see the previous results, the first results. Luckily, our most preferred route, which was the bath, which you can see here in the black lines, was the one that was causing the highest cumulative mortality. But what was even more interesting is that if we not only used a relatively high dose of virus in the water, but we also extended the exposure time of the fish to the virus, then we actually had almost 100% mortality in this young fish, which is exactly what we wanted to obtain. So a prolonged exposure of young fish has been chosen. So the bath challenge with prolonged exposure has been chosen for any of our subsequent experiments. Then we also had to look at our window susceptibility, so at the age sensitivity. And for that, we used the same carp strain provided by Wageningen and using the challenge model optimized previously, but this time we exposed fish at different age stages just to check what was the window of susceptibility of our own strain. And then, as you can see here very nicely, there is a very strong age dependency, and this experiment allowed us to confirm that our age or window of susceptibility is between 3 and 6 to 7 months. And this is very important because, of course, that meant that we could not challenge the fish any later than 6-7 months, because otherwise it would become naturally resistant and we would not be able to distinguish the effect of our vaccine from the effect of the natural resistance developed by the fish. So now we had a very nice challenge model. We knew when we had to vaccinate the fish, so we came up with the following scheme that we followed throughout all subsequent vaccination and challenges that we performed at Indra. So we always had the hatchery manager in our facility, Manotte Veld, always communicating with Dimitri at the hatchery facility in at Indra. They communicated about the production of the eggs. The eggs were sent from Wageningen to Indra within a day. Then they were raised and grown at Indra for 3 months at optimal growing temperature between 20 and 22 degrees. Then, with the help of the team at Indra, we performed the vaccination. And then we were always very, very strict to not challenge the fish before 2 or 3 months after vaccination. And why was that? Because the first vaccine that we tested, as I will show you in the next slide, was a DNA vaccine. And as many of you may know, even just the backbone of the DNA vaccine can actually cause a kind of aspecific protection due to very high upon interferon production. So to make sure that the protection that we would see would be really conferred by the specific immunity triggered by the vaccine, we always allowed a very good window, very good time between the vaccination and the challenge. So 2 to 2.5 months was the earliest that we ever vaccinated that we ever challenged after vaccination. And then to also be as close as possible to the natural conditions during which the fish are most susceptible to the virus, we also slowly, a few weeks before the challenge, lowered the temperature at which the fish were grown to 15 degrees, which is, let's say, the optimal temperature then for the virus, or the temperature which the virus usually causes outbreaks. And then the challenge was performed, of course, always at Indra. And in between, we could join to perform sampling, to if we had to take serum or if we had to take organs. So let's see how this worked out for our first trial, which was a DNA vaccine. So we have a DNA vaccine encoding for the glycoprotein, as I told you before, of the SVCV virus, and we tested a, let's say, a relatively high normal dose for a DNA vaccine, but also a 10 times lower dose. As mentioned before, the vaccination was performed at 20 degrees, whereas the challenge at 15 degrees for a prolonged period of time at the optimized dose, as I showed before. And as you can nicely see here, in the dashed line are our controls, so the empty plasmids, and in the full line up here, you have the, let's say, survival of our vaccinated fish. So you can see that both at 1 microgram or even 0.1 micrograms per gram of fish, we could confer up to 100% protection to common carp. So we actually were quite happy because even 0.1 micrograms were very, very effective. So as I mentioned before, vaccination and challenges were done at NRAM, but at the same time in Wageningen, we could also just simply grow exactly the same batch of fish, vaccinate them exactly with the same batch of the vaccine, but we could also sample the fish in Wageningen and perform immune response analysis in parallel to the challenge experiments that were done at INRA. So I will of course not show you that part because that's not part of the use of the facility, but still I wanted to say that it led to at least 3 nice publications, of which a couple I will mention here. So the first, where we basically fully characterized the specific immune response triggered by this DNA vaccine. And then the next one was, of course, now we have this very successful DNA vaccine that needs to be injected. But if you think that there are 20 million tons of carp produced every year, well, you don't want to be the farmer that has to inject every single carp in their farm. So of course, as many other, let's say groups are trying to optimize, we tried to convert this injectable DNA vaccine into an oral DNA vaccine. And what did we do? So again, we took 3-month-old fish, and this picture is just to show you that, of course, a 3-month-old carp is a very, very, very tiny carp. And this is just to show you the tip of a 10 microliter tip. So you require quite some skills and quite some training to be able to very quickly orally vaccinate or intubate fish, at least for these first experiments that we performed. Then we used 2 different doses, one that was 3 times of a low dose and one that was 1 time of a higher dose of the DNA vaccine. And of course, to protect the plasmid, we encapsulated into so-called alginate microsphere. Then, as before, we challenged after 2.5 months with the optimized challenge protocol. And here, of course, we use our injectable DNA vaccine as our golden standard. So that one conferred almost up to 100% protection as expected, whereas all of our oral combinations were not as protective. And some of them were even performing worse than our control group, but I will come back to that later. So then we thought maybe the dose of the vaccine is not too high. Based on other experiments performed in trout, for example, where they were partly or partially successful in protecting trout against IH and V infection using an oral DNA vaccine, they use much higher doses than we were using here. So in the next experiment, we did 2 things. We increased the dose of our DNA vaccine, but we also added a mucosal adjuvant. And a mucosal adjuvant that is actually also normally used, for example, for mucosal vaccination in mammals or other veterinary species. So we used 20 to 100 micrograms of DNA per gram fish, so a much, much higher dose. And then the rest was the same, vaccination at 20 degrees, challenge at 15 with the optimized protocol. Again, we have our golden standard here, our intramuscular DNA vaccine always working very well, but unfortunately, at all the combinations, the oral vaccination did not protect. And then, as I mentioned before, we were worried about certain groups because some groups that were orally vaccinated with a relatively high dose of the plasmid, here, 100 micrograms, even in the presence of a mucosal adjuvant, they were dying faster. So we took a side step. So the mucosal, unfortunately, up until now, the mucosal oral vaccine, even in the presence of adjuvant, was not yet successful. So before we proceeded or went on in further optimizing our oral vaccines, we took a bit of a sidestep and we asked ourselves why some of these groups are either dying faster or even dying a little bit more. So there were more fish, not surviving the infection. Could it be that the oral vaccine triggers tolerance? Which is something that definitely we don't want, because tolerance would mean that once the fish would be exposed to the pathogen, then they actually would perform even worse. So this is one of the last experiments that we performed. And then again, we used our intramuscular DNA vaccine as our golden standard. Oh yeah, now I have my cat that is really interested in the presentation. So we have our intramuscular DNA vaccine as our golden standard. Then we gave our oral DNA vaccine as shown before, but the low dose. And then we had two groups. One where we first provided the oral vaccine and then we intramuscularly injected the DNA vaccine. And this is the most important group to tell us whether oral vaccination prior to intramuscular DNA vaccination would confer tolerance. And then we had another group where we first performed our intramuscular DNA vaccination and then gave an oral vaccine to see whether there would be a reduction in the efficacy of our, let's say, golden standard. So, and again, most important, to allow enough time for either tolerance or protection to occur, we always kept at least 2 months between the two vaccination period and of course between the last vaccination and the challenge. So does oral DNA vaccination or oral vaccination induce tolerance? So here again we have our golden standard. So we have our DNA vaccine and the control, so that also works always nicely. The oral DNA vaccine did not confer a lot of protection. In this case, the fish were dying not faster, but at least there was no protection as expected. And this is the most important group because this is the one that first received the oral vaccine and then the DNA vaccine intramuscularly, and we do not see any reduction in the protection. And also not when we first intramuscularly DNA vaccinated and then provided the oral vaccine, which gave us good hope that basically the problem is in the optimization of the dose, of the adjuvant, and what we think now most importantly of the regime of the oral vaccination, and not because oral DNA vaccination may confer tolerance. And this was actually a quite important step to take because all other subsequent experiments that we're currently performing are still aiming at improving or actually turning our successful injectable DNA vaccine into an oral DNA vaccine. So indeed, we have been testing also insect cells-based tribunal vaccines, both by injection and orally. We are now currently busy with a plant virus-based VLP vaccine, also lactococcus-based, so using commensal bacteria to deliver DNA vaccine. And all this were basically done just before Corona. So we are actually waiting. Unfortunately, we could not analyze the samples, not all the samples, to actually be able to decide what to do next. So I think since the start of, or the end of AquaXcel and the beginning of VetBioNet, we have had at least 5 visits to the facility. So, and I have to say that we are extremely happy with the service, with the possibilities, with the communications and arrangements that are done and can be done with the team, with Dimitri and all the other colleagues, with Damian, Megali, and Philippe. And yeah, as I said, I had to make a selection, but I hope that this gave you a little bit of an overview of the possibility that this can be that are possible when it comes to carp vaccination challenges in the facility at INRA. And I would like to thank you for your attention.
Thank you very much, Maria. It was a nice presentation on the possibilities of vaccine research within several EU projects. You were referring to AquaXcel, which was a preceding project, and I think this is also, I hope at least, in the sense of the EU funding, that projects are linked to each other, and that there is a major, that they will take a benefit of cooperation across projects. First of all, are there any questions to Maria in regard to the scientific content of the presentation? Please feel free, but you have to unmute your microphone. If that is not the case, I have one question to you, Maria. You choose purposely for a very long, for at least in veterinary terms, a very long period between the vaccination and the challenge. And we often have this discussion if we are working in biomedical research, that the biomedical research for human vaccines, they also would like to have this long interval. For veterinary purposes is often very difficult because our animals are exposed and we will have them protected as soon as possible. How is it with carps? You were referring to the disease, that it is that the highest mortality is in the young age. So isn't it strange that you work for a vaccine which is protecting a life which is not any more susceptible, or less susceptible, or getting less susceptible?
Yeah, I know what you mean. So you say if the adult carp would become naturally resistant, why are you challenging? The point is that we know that not all carp strains, and actually not all individuals within the farm, are naturally or becoming naturally resistant, and that's actually the major problem. It's there is a bit of a individual genetic predisposition that, of course, makes so as far as I know, there is not one strain that is absolutely fully resistant to SVCV. So that's why, for example, we have this mix of R3, R8. The R8 alone is generally more resistant, but still you may get 10 to 20% death. And then all koi carp that I know, so the ones that are really used for shows and are for ornamental purposes, they're all really, really heavily susceptible. So I think if you look at the variation of carp strains that are there, I think it's still worth it to have the possibility to have a vaccine to basically prevent, rather than being faced with the fact that maybe one farm has only 20% loss and the other farm can actually still have 60, 70% loss, even though they have adult animals.
Yeah. Okay, thank you.
And then another thing is that, of course, if you look at the hatching period of carp, they hatch in May, and that's around the period where actually the virus, so you really want to help them. So they hatch, and then you want to wait 2.5, maximum 3 months, you want to vaccinate them, and then that's the period where they're still susceptible to the virus. So if you look at the real practice, then the vaccination period is actually perfectly timed for their susceptibility period.
Great. Okay, thank you very much.
Yeah.
Monica,
I have a question, if I may. You're talking about mortality and high percentages of mortality. Does that actually mean that it is death as an endpoint in your experiments? Or do you have humane endpoint criteria that allow you to pick the fish out that are demonstrating clear signs of disease?
Yeah, of course, it's the second one. So we don't wait until the fish, let's say, flips open or flips up. No. So Dimitri and his colleagues all very well trained to see when the fish becomes lethargic, when the fish doesn't or eats less. That's the point where we consider the fish that can be removed from the experiment before, of course, it becomes too painful.
I see Donica has a question.
Thank you, Norbert. Lovely talk, Maria. I wondered, at the start you said we don't vaccinate young fish because of their immature immune system. And I can see that reasoning, but have you ever tried it? Because in the chicken, where I'm active in, we vaccinate in ovo with more than one vaccine. And it's got a terribly immature immune system, but it works unbelievably well. And we don't know why, but it does work. So have you tried it all in the fish?
Yeah, so this goes back to maybe experiments of Winkel or something like 25, 30 years ago, where they did try to vaccinate carp and also trout in an age period or weight that the fish were between, let's say, 0 and 2.5 months, let's put it that way. And then when they were exposed to a bacterin, which was at the time, the golden standard for the vaccine against Aeromonas and also Yersinia, then the fish were more susceptible to the infection than if they were vaccinated after the 2.5, 3 months. So there are very early studies that showed that age and weight are important for young fish to develop immunity.
I realized those studies. I think it was the time I was studying at Wageningen. But the point is, at the moment, A, the fish have genetically changed, and B, you're not using a bacterin. You know, your vaccines are completely different. Your delivery systems are completely different. I don't think you can draw any conclusions from the studies done 30 years ago, to be honest. I'm surprised you haven't tried it.
I think the companies are very much, at least I know for salmon, companies have tried to vaccinate salmon, let's say smaller than 1.5 grams, and then they get the same. So because of course they have to use less vaccine, both by IP, by immersion, anything. And then again, you don't get protection. So those are more recent, let's say, studies, but they are not published just because this comes more from the practice. So at least from salmonid, again, we know, but in that case, it's based on weight. But you reach a particular weight only when you have a certain age. That then again, if you vaccinate too early, you do not get protection.
And another question is, have you looked at the expression levels? So when you intramuscularly supply the vaccine or orally, so what are the levels of expression in the end of your protein of interest?
Yeah, so the SVCV protein is extremely high expressed, but what we do know is that it triggers a massive type 1 interferon response, and we have 4 groups of type 1 interferon in fish. So they're all very highly upregulated when we perform the intramuscular injection, but the fold changes drop dramatically when we do the oral vaccination. So although we know that the plasmid is perfectly delivered because we can detect using antibodies against the protein, we can detect the expression of the protein in the gut, we still see, compared to the intramuscular injection, much lower fold changes of many of the immune genes that we now know are associated with protective intramuscular DNA vaccines. They are much lower during the oral vaccination. We now think that it's really the lack of a proper immune stimulation when we give the vaccine orally that is the problem.
That's a very common problem. Thank you.
Thank you very much.
Yeah,
it's a very interesting discussion on it's a very interesting discussion on the immunology of the different species, and I would like to stress that also immunology questions are very often addressed in research projects or in TNA projects, either in pig or in chicken, and I can really encourage researchers to take the to look for the expertise which is available in VetBioNet on this specific part. Maybe one question to the colleagues at INRA who joined and who supported Maria in this study. Just for curiosity, what does it mean to have a TNA project in your facilities? What does it have in organizational terms? What does it mean for you? Bertrand.
Yeah, I can say a few words, but probably Dimitri and Pierre might add as well some comments.
Great. Yeah, they're also here. Yes.
Well, I think it's a great. I mean, TNA is a really good way to actually foster some, to reinforce some collaboration that we already have, because of course in the fish scientific community we are a small community. We know each other well, I think, And it's the TNA is a really interesting way to actually to develop an idea and to develop and to test an interesting hypothesis, for example, for a new vaccine or a new so it's yeah, for this it's extremely useful. And so there's many examples of these, and I think with the projects that are coming up in our facility in Rail, we are going to again, you know, hopefully do some good work on these with colleagues. Yeah.
Great. Pierre, you want to add something?
Maybe I can add that it's always interesting actually to confront our usual way of working with our colleague of the fish facilities with the culture of another research group coming with their own way to interact with fish and with their own facilities. It's actually very, very interesting to exchange on this. And as Bertrand mentioned, if it's with a group that we already know, it's even easier because the communication is, of course, very easy and straightforward. So it was extremely positive.
Thank you very much, Pierre. I think you mentioned something which has been proven in the past and also all the time. The communication skills of the service provider and of the user are very important in having a successful project. I think this is very, very important. And but this most of the time, because we are talking this is the same scientific language, it makes things quite easy and easier. Maria, I would like to thank you for your presentation and would like to go forward to the next presentation by Llilianne Ganges from CReSA. And Maria, you have to leave the, yes, and Llilianne, if you can activate the screen, and in between I will tell a little bit about the project. It's Llilianne will talk about the project from Anna Maria Moreno from Isla in Italy. But Anna Maria was not able to be present today, and Llilianne is so kind to present the results, and will have the double function of being a service provider and giving the information by the user. But I think she will manage it very well. Llilianne, welcome, and the floor is yours.
Thank you very much for your presentation and introduction. So it's more easy for me now. I try to chat in again.
Yeah, you are in.
It's okay? Can you see the screen? Okay.
Can you a little bit increase the size?
Okay, maybe in the
presentation.
Okay.
Yeah, excellent.
Fantastic. So, okay, as according to your presentation, I am now the W functions, but this was applied by Dr. Anna Moreno from Isla in Italy to perform this transnational access activity project about the experimental infection in ewes and pigs with a novel Pestivirus closely related to classical swine fever virus that we performed in our facility that at the same time is an OIE reference lab for classical swine fever. So, okay, sorry. The Pestivirus genus is important to consider that it belongs to the Flaviviridae family and has been described at least 11 species that were recently classified from the Pestivirus A until K. However, in the last 5 years, several Pestivirus has been also recently described, including the atypical porcine Pestivirus that can infect pigs, and also other in whales and even in pangolins, and including this new Pestivirus. And really is a big concern the emergence of new Pestivirus species, considering the high capacity of adaptability of this virus and the broad host range. So, okay. It's very well known that the Pestivirus infection in the different hosts has the capacity to produce a range of clinical conditions characterized by acute diarrhea, acute hemorrhagic syndrome, also the acute fatal disease, Wageningen disease, and also the subclinical case. And the Pestivirus infection has the capability to be transmitted by transplacental transmission. And the transplacental infection is a very dangerous for because help the prevalence of the Pestivirus in the nature because it induce and generate congenital animal born persistent infected can help to increase the circulation of the virus worldwide. About this new Pestivirus, the evidence was found in Italy in 2017. The college from the ANA lab isolate a new Pestivirus that was isolated directly from aborted fetuses, and then also performing the complete sequencing of this virus and performing this analyzing to that prove that the new Pestivirus isolated from this species was very genetically close with the classical swine fever clade. However, this new Pestivirus was aggruppated in a new clade specifically. Okay. Is important to consider that this close genetic relationship that was found between the novel ovine Pestivirus with classical swine fever virus is a great concern, because classical swine fever is a notifiable disease to the World Organization for Animal Health, OAE, and consequently, the infection, the probability of this virus can infect pigs, is really necessary to rule out. And furthermore, the pathogenesis that this virus can induce in the host, in the ewes, the natural host, as well as the probably abortogenic role also in this host, as well as the pathogenesis that the virus can induce in pigs, is also need to be clarified. Sorry. So the objective of this project was to investigate the host range of the novel OBPV, in particular the capability to infect pigs and the development of clinical disease in the pig species, as well as to clarify its pathogenic role by inoculation in pregnant ewes in order to investigate the capacity of the new OBP ovine Pestivirus to generate vertical transmission and abortogenic role. And finally, to investigate the potential cross-reaction between the new Pestivirus with other Pestivirus previously described, mainly with classical swine fever. Sorry, because, okay, we performed the experiment, and really we working very hard to carry out the both experiment, the in vivo experiment using pigs and also pregnant ewes in our facility. Therefore, we don't find any difficult problems to develop that. However, we need to working hard to go out to develop the experiment and the transnational access activity. So we performed the ethical protocol because we need to present our procedure to the ethical committee for obtain the approbation, and according to the ethical for animal experimentation rule, and we obtained this permission from both experiment in both species. And the first experiment that I want to present was developed in pregnant ewes that was detecting Pestivirus free, and also from other bacterial infection that can induce abortus. And at 63 days of gestation, we allocated the pregnant ewes in our facility in the Biosafety Level 3 lab, and the animal were inoculated with the isolate, the new Pestivirus isolate, using 5 per 10 to the 6 infectious dose, considering other previously described experiments with border disease virus in this species. We proceeded with the daily clinical evaluation during 84 days post-infection, about 144 days of gestation. And we collecting different samples to monitoring the immune response and also the viral replication and excretion during the experiment until the end of the study. And also we evaluate, we contemplate to practice the euthanasia in the case of some animal presenting abortions. And the animal that aborted during the study was euthanized, of course, and collecting different tissues to evaluate the viral replication capacity, including the placenta and also in the foetuses, all these tissues that I explained here. Sorry, it's a little delayed. After the infection during the experiment, in this image, you can see here that we detecting that 5 of the 8 sheep aborted during at different time of the study. Only two animals presenting premature birth with dead lambs, and the only one of the sheep gave birth to a lamb that was born with clinical sign. I was presenting after the 2 days after born, developed conjunctivitis, fever, and polyarthritis, tremor, and we finally practiced euthanasia in these animals. So we prove here that really the new pestivirus can induce abortogenic problems in these animals. I have a problem with the, sorry. Okay, here we show the replication, the capacity.
We lost the presentation, Llilianne.
Sorry?
We don't see the presentation, at least I do not see.
Oh my God, sorry. I don't know what happened. Okay, now?
Not for me. Anybody?
No.
I think you need to share your screen again.
Don't let me sharing with you.
Llilianne, a suggestion: stop sharing and start it again. It might work.
Sorry now?
Yes, excellent.
It shows up.
Yeah.
Oh, sorry, my apologize. Okay, I tried to explain now that here we show the capacity.
Sorry, Llilianne, please switch in the presentation mode, if possible.
I don't understand. What do you need?
In the presentation mode, the screen of the PowerPoint.
Sorry, it's because I have a problem with the.
Okay, go ahead. Go ahead.
Yes, now?
Yeah, excellent.
Great.
Sorry for that.
No,
okay, now I only want to explain that, okay, we detecting the viral replication in the sera and in all the animals that were infected using the Pestivirus real time. Also, we developed a new real time to monitoring this virus. Also, the interesting here is that for the first time, we detecting one pestivirus that can react in the molecular diagnosis with classical swine fever, a specific assay that is the official for the OIE detection of this virus. So this OBPV can interfere with the molecular diagnosis, and we detecting that the animal were positive for classical swine fever also real time. So after 60 days post-infection, we detecting also the presence of the virus in the plasma of all the animals, and also the persistence of the virus in the tonsil for at least 2 months after infection, including, of course, with the cross-reaction with the classical swine fever molecular diagnosis that you can see here in this blast. Okay, so this is the problem. Very big problem for the classical swine fever official diagnosis. So also in the foetuses and all the dead lambs, we of course the same detecting the replication of the new new ovine pestivirus that we say OBPV, and also cross-react strongly because the viral load is higher, so higher in the animals, in the foetuses, and also in the lamb that born 6. And also with at the same time, described here that the new ovine pestivirus can pass by transplacental transmission from the infected egg to the lambs, and the virus can induce persistent infection and develop some disease in the infected lamb that born with this persistent disease. In the case of the serological test, here in this panel in red color, you can see that we detecting the specific antibody response to classical swine fever virus very fast after at the 7-day post-infection that was increased in time, at the different with the humoral response that we evaluated with the ruminant pestivirus ELISA. The ELISA for classical swine fever virus is more specific to detect the humoral response to the new OBPV virus. In the case of neutralizing antibody response, also we studied this and we characterizing that. And as you see in this table, I don't know if you can see this. It's very sorry. All the animal infected, this is the homologous virus, the response, the neutralizing titer, again, the homologous virus that really is very high at 34 days post-infection, and also at 41, but it's impossible to differentiate the neutralizing antibody response with classical swine fever virus. This red part that I highlight here represent the different classical swine fever strain that we use to differentiate, and the immune response, the neutralizing immune response is similar and is very difficult to differentiate with the OBPV, with the classical swine fever strain from genotype 2.3 in specific. To understand this high cross-reactivity found in the neutralization assay, we tried to analyze the antigenic relation between the OBPV and classical swine fever and other representative pestivirus strains, and we developed this study that was included in the manuscript, in the paper, and we proved that the classical swine fever strain from genotype 2.3 has the more high antigenic relationship with the new OBPV than other strains from pestivirus that we using in this study. Also, we performed one study to understand the evolutionary history and co-phylogenetic reconstruction to understand the emergence of this OBPV and the relationship with classical swine fever and other pestivirus. And we found after this very deep study that the OBPV and the classical swine fever sharing the common ancestor is another pestivirus, that is the Tunisian pestivirus that probably emerging around 300 years ago. And this study is suggesting that the differentiation of this Tunisian pestivirus into two dangerous new virus from animal herd, classical swine fever and also the ovine pestivirus, that was likely favored by human intervention at that moment for the close housing of multiple species in that 300 years ago. And this is all that we can perform in this study. To cover the other objective of this project, we also developed the experiment to investigate the host range of this novel ovine pestivirus in pigs to understand the capability of the virus to infect the species, considering also the high cross-reactivity that has not only in the antiviral response, also in the genetic and molecular diagnosis. So the experiment has carried out without any problems, and we proving that the virus also have the capability to infect swine, and all the results are now under analyzing for publication. So at the moment we don't sharing this information. So that's all for my side in this project. We will discuss maybe in other meeting this when we obtain the publication. So thank you very much for your attention.
Thank you for your contribution, Llilianne. It looks very promising, and I'm pretty sure that in a following meeting we will be very curious on the results in pigs. Are there any questions from the audience to Llilianne? Maybe one question from my side, Llilianne. I didn't. Maybe you mentioned it, but what is the prevalence of this virus in Italy or in Spain? Did you look for it already, or was it more an incidental finding, and you wanted to rule out the possible danger for the livestock?
At the moment, we prove that it's danger for the ovine species, because really the virus can induce severe abortogenic role in this species, because from 8 animals that we included in the experiment, really 100% of them developed some problem. The majority of the infected animal abortus, presenting abortus, mummification, and the less amount of the 8 from the to 8 presenting premature birth with dead animal, and the only that was capable to give a viable alive animal, the animal born with really serious problems, weak. Really,
I don't understand this from your presentation. My question was more, what is is there anything known about the prevalence of this virus in the field infections?
We need to know. We need to perform very deep study, because the problem is that we don't have the OBPVs isolate to including in the panel of, anybody analyzing, studying the humoral response in sheep using the ELISA of classical swine fever, because it's other virus species ELISA. So it's very difficult, because anybody has in the mind to use one system to diagnose one disease in pigs using in the sheep species. And at least in Spain, this species is less studied than the swine, and maybe really is we discussed it in deep in the paper that are at least 50% of abortus cases of sheep that are misdiagnosed. And we need to use maybe the ELISA of classical swine fever virus to study the prevalence, at least in these species, to know and to detecting antibody response, and also studying the Pestivirus by Pestivirus molecular assay to detecting and studying the abortus case in sheep. And in the case of pigs, also is a problem for the diagnostic, because maybe we can see, we have in our mind that we have a border disease case, because this virus can cross also with border disease. And sometimes when we detecting by the ELISA of classical swine fever, by using the monitoring programs for classical swine fever surveillance, we detected some cases E2 positive animal, by the ELISA of classical swine fever, but we don't have the OBPV, the new emergent virus, to including in the panel that we use in the seroneutralization test to discriminate, Of course, at the moment, this disease is not under study, and we need to use the tool that we have, and we are an OIE reference lab for classical swine fever, and we reivindicate in our meetings the study more in depth, the prevalence of new pestivirus and emergent in different, using this kind of approach at the moment. Okay.
Any more questions? I don't see any. In regard to your service, the service you had to provide within the Transnational Access, I understand that this is already, again, a cooperation which already existed and which is proceeded in the Transnational Access. Is that correct?
Yes.
Yes, and that is something which we see very often, that we support colleagues with the Transnational Access, of which we already had cooperation with, and which need to do and to proceed with certain and specific studies. I think this is a very nice example of the veterinary community in cooperating together in looking for this type of emerging pathogens, which is a quite important part of the high containment units and the high containment work of VetBioNet. And really thank you for this presentation, Llilianne. And I would like to close this this particular part of the afternoon public not without giving my thanks to the user selection panel. Just for those who are not familiar to the process, the proposals for a TNA study are carefully reviewed by an external user selection panel, experts who are not part of the VetBioNet project, and they look carefully in the design of the project and the research question, and they advise the executive committee of VetBioNet on the scientific background and the scientific quality of the feasibility of the project, and VetBioNet decides on the giving the grant to the specific project. So many thanks to the members of the USP who are some were in the audience, and I saw that some have left in between. But I think it is important to thank them on behalf of the VetBioNet consortium, because they put quite some time into the project and the project evaluation. And although I think we all enjoy these discussions, they also take time in preparation and in being available. So many thanks to the USP. With these words, I would like to give back to Sasha.
I've just seen there's one question in the chat, Norbert. Perhaps you can respond directly. It's a question from Monica from EVI concerning services. Perhaps you can directly exchange about.
Monica, you're a little bit far away. Please go closer to the microphone.
Is it better now? Is it or not really?
Yes, I know you're not well. I cannot hear you well at the moment, but I have your question here, and please confirm that your question is in regard to providing services to collaborators or partners from the project. And yes. This is the point is that the project, that the Transnational Access Project is always submitted by a research group from outside the consortium. That does not mean that collaborations or links between the request, the applicant, and the service provider are already present. Our field is so small; it's nearly unavoidable to have this, and I think this makes it, on the one hand side, difficult to not to have a link in one or the other way, and on the other hand, it is an advantage to address questions from colleagues outside VetBioNet who are still very urgent or important, and which can be worked on during the in the VetBioNet consortium.
So there's no problem with having the partners from, let's say, SWR applying for the service at INRA, because we got the comment from the European Commission that for Transnational 2 we should avoid applications from partners that are in the consortium.
Yes, of course.
But although we know that, for example, if maybe INRA has different groups, and only one of them is in consortium and others not. But okay, this is coming from
no, but the rule is that we never have a responsible researcher research group. Or the research group which is responsible for a project is part of the VetBioNet consortium.
Okay.
Okay, so I